this lecture series, and especially tonight's lecture, is supported by two very, very kind gifts to the program. One is for the Claire Matzker-Lilienthal Fund, which supports lectures in science and the wonderful topics that we have here this evening, as well as the Myers Family Fund. So I'd like to make sure we acknowledge both of those donors. So, you know, when you're asked to introduce folks to a program like this, they give you all this kind of standard biographical information, and I'm going to ignore it all, as it turns out. But we're going to have a fascinating lecture tonight between Katie Pollard and Shannon Bennett, two really wonderful scientists who will be talking about the little things that make the world go, especially our kind of microbiome, the things living in our bodies, and why bacteria turn out to be our best little friends. So that's going to be really the topic this evening. And I was kind of quizzing our speakers tonight about interesting factoids about them. I'll give you the official version and then the interesting bits I gleaned this evening first. Katie Pollard is a professor at the University of California, San Francisco. She's in the Department of Epidemiology and Biostatistics, and she works on human genomics. And she's also a fellow at the Gladstone Institute and a fellow at the California Academy of Sciences. She's one of the world's leading authorities on the human microbiome, again, the living things inside our bodies and how we can use modern genomic tools to kind of understand all those critters living inside all of us. But, you know, and I had pages and pages of all of her scientific accomplishments, but what I was really fascinated to find out is that she was in an all-female punk rock band earlier in her life. So I think that's really cool. Much more interesting than her CV. And then Shannon Bennett, who I'm very fortunate to know is a colleague at the Cal Academy, is one of our rock star scientists and curators at the Cal Academy. She's our very first curator of microorganisms at the Cal Academy. And she's also a Patterson Scholar, newly named at the Cal Academy, for some of our most preeminent scholars at the Cal Academy. She focuses on viruses and bacteria and the relationships between what's happening in the environment and the diseases that we can get through the environment. So links between biodiversity, disease, and human biology. So fascinating topic. And I also, though I've known Shannon for months now, did not know that in her previous life she also learned how to be a chicken hypnotist. So go figure. So we have a punk rocker and a chicken hypnotist coming out this evening to talk about the little creatures in our bodies that outnumber us about 10 to 1. So it should be pretty interesting. So let's give them a big round of applause as they come out here to speak.
I'm so lucky. Chicken hypnotist? That sounds like a punk band. I think we need to collaborate. Good idea.
Well, it's great to have you here, and I think I can speak for everybody in the audience that we're looking forward to an evening teeming with microbes. We really want to be grossed out this time. So, Katie, your work has obviously advanced a lot of our understanding and appreciation of how many microbes really make us who we are and all the various jobs they do. So I would really like to kick off the evening with some of the basics. How many microbes make up the human body?
Great question. Actually, to be honest, we don't know really for sure. And frankly, we actually don't even know how many human cells are in the human body. But let's just go with approximations and try to talk about it. So first of all, I think we need to define what a microbe is. So when I say a microbe, one of the first things that comes to mind is bacteria. That's in the title of the program for tonight, and bacteria are a big component of what's living in our bodies, but there's other little critters there too. So there are another entire phylogenetic group called archaea. They're also single-celled organisms. In some ways, they're more closely related to us than bacteria. They're also single-celled. They're very diverse. They're as different from bacteria as they are from us. And they're best known for things like living in the hot thermal vents in the bottom of the ocean, but there's actually quite a few of them in our body as well. On top of that, we have microbial eukaryotes, so organisms that are very closely related to us, but they're single cells. Probably the ones you know the best are things like yeasts that we use for bread and beer and all kinds of good things, but also responsible for things like athlete's foot or other kinds of infections. And there are others, parasites, definitely parasites. And you don't want to forget about the viruses. Very small. Shannon's specialty. They're very small. They're not really cellular organisms, but we're going to include them with microbes tonight. They have genetic material. They basically hijack our cells or cells of bacteria to promote themselves. So they're part of microbes. Okay, so we got bacteria, archaea, microbial eukaryotes, and viruses. Now, how many are there? Great question. So, we're not even sure, as I said, how many human cells there are in the body. But let's just go with approximations and say between the humans and the microbes, there's about 20 trillion cells. Okay? And then I thought maybe we could engage the audience a little bit and try to vote by clapping for what percentage of those 20 trillion cells you think are microbes. All right, and we'll make it multiple choice. We'll make it multiple choice, so it'll be fun and easy. You can win just by guessing. Okay, so 1%? Wait, 10%, 25%, or 50%. So those are the four choices, and we're gonna clap. So there's 20 trillion cells, is it 1% microbial? Clap if you think that's the answer. All right, 10%. Yeah. 25%? Option C is always a safe answer. Okay, 50%. All right, you guys are smart. So it's probably at least 50%, and by some estimates, actually, it could be 90+% of the cells in our body are microbes, but it's at least 50%. So I said, let's assume there's 20 trillion cells. Let's just conservatively say 50%. That means there's 10 trillion microbes on each one of us right now. That's a big number. That's a big number. So what's 10 trillion? You might be familiar with the US debt. It's about the same order of magnitude. Maybe that makes it seem small, I don't know. What about the number of galaxies in the universe? That's about the same order of magnitude too. So there's as many microbes as in your body right now, approximately, as there are galaxies in the universe.
Incredible. We are almost not even what we think of as human. We're more other than we are human.
We are, and that's not it. I'm talking about cells right now. Let's talk about genes. The story gets worse. Okay, so a little biology lesson. So every cell in your body that's human, we're going to call it the human cells. Really, they're all human, but the ones that have human DNA in them that you got from your mother and father, every one of them has the same genetic material in it. Modulo, what we call somatic mutations that cause things like cancer, but most of the cells have the exact same DNA. So out of the approximately 10 trillion cells that are human, you have the same genes, and there's maybe, again, scientists aren't really sure. We're very bad at getting these numbers accurate, but let's say on the order of 25,000 genes. But they're the same in every cell. Now, there's another, let's be fair, just conservative and say 10 trillion cells that are microbial, but they're of thousands of different types with different genetic material. And even though their genomes are a little bit smaller, maybe 5,000 genes instead of 25,000 genes, that means by very, very conservative back-of-the-envelope calculations, there could be 150x as many microbial genes as human genes in your body.
So in terms of the activity that's going on, the genetic diversity and activity of the microbes far outweigh what our own cells are doing.
Exactly, and genes make proteins, and proteins make cells and do all the things that your body does. And so if most of those are bacterial, then studying the human genome is really only showing you a small part of the picture.
The tip of the iceberg.
Yeah, okay, lest anyone's totally freaking out right now, the microbial cells are smaller. And so by body weight, actually, you are mostly human. About 6 pounds of an average person's body weight is microbial.
Huh. So we outweigh them.
Do you feel better now?
A little bit better, but not much.
So about 5 of those pounds are in your gastrointestinal tract. That's where a lot of them live, but they're actually in most of your body. And we used to think a lot of organs were sterile. Medicine has taught us a lot of organs are sterile, but the more we look, we find microbes in all kinds of parts of the body. And so even though the bulk of them are in our GI tract, and we're going to talk a lot about that tonight, keep in mind it's also your skin, your ears, your nose, your lungs, your blood, your immune system. Some even get into across the blood-brain barrier.
Wow. So it sounds like there are really a lot of microbes in our bodies, and they're doing a lot of things with all the different kinds of genes they represent. Do we know what they're doing there?
Mostly not, actually. So I can tell you a few stories, and we're going to talk about some of them tonight. And every day, literally every day, there's new evidence coming out for what these microbes are doing. But frankly, we're, as you said, tip of the iceberg, just beginning to understand this. And the reason is that most of the microbes that live in the human body are not what we call culturable or cultivatable. Traditional microbiological methods could not study them. You couldn't take them out of the human body, grow them in a dish in the lab, look at them under the microscope, subject them to experimentation. They didn't survive outside of their normal ecosystem and without their microbial partners there, the interactions with one another on which they depend. Right. So for a long time, microbiology has really focused on a very small part. It's estimated about 1% of the microbes that are known have really been carefully studied. So we're expanding that, and we'll talk tonight about some of the techniques we use to get beyond that little 1% to try to see what's there, which is really driving amazing discoveries right now. So let me outline a few of them. What are these guys doing? So we are mostly aware of pathogens, and have been for a long time. And they're important, and they're there. So I can bet that pretty much everyone in this room, and everyone who's listening, has at some point said, oh, did you get that flu virus this year? Did you get the vaccine for flu? Or, oh, I'm not feeling too great, I think I have the flu. So we're very familiar with flu, one tiny little virus. It has a big impact on our lives that we're very aware of. But I imagine very few people have said, wow, I feel like I don't really have the right balance of firmicutes. Have you?
Never.
Maybe you should have. These guys are really important. They make up the bulk of the microbes in some people's GI tracts, and they're present in all of us. And some of them are good, and some of them are bad, and knowing which ones you have and what your balance is can really make a big difference. So what I'm trying to say is that pathogens are important, but a lot of the microbes that live in and on us are what we would call a commensal. They are either just hanging out, they're along for the ride, or in many cases, they're an integral part of our normal biology. They are part of us. And what happens to us, how we respond to a drug, what happens when we eat food, whether we're at risk for disease, has to do with what these microbes are doing and how they're interacting with each other and how they're interacting with our cells. So digestion is one of the major areas. As I mentioned, most of the microbes are in our GI tract. So what the heck are they doing there? We don't really know, but a few things are becoming evident. They do things that our cells can't do, like make vitamins that we can't make, break down certain things in our food, like certain fibers that we can't break down. They metabolize drugs that we take. So they are helping those drugs become bioactive in our bodies. So when we see in a trial that some people respond to a drug and some that don't, or other people have adverse effects, it may be not their human genetics that's driving that, but the genetics of their microbiome. Specifically, fiber is really important. So this is not digestible by humans. Bacteria, certain bacterial groups take fiber, do digest it, and they make something else, which is called a small chain fatty acid. Most people might be familiar, maybe you've seen SCFA. It's also what happens in your armpits when you get body odor. So these small chain fatty acids are the reason for deodorants, but they're also, more importantly, what's happening when you eat anything with fiber in it, any kind of plant food. So why is that important that you're making these small chain fatty acids? It's important for the microbes because the presence of them signals to our immune system, don't attack these guys, they're good, don't mount a big immune response. It allows that 10 trillion cells to exist in your body without you being in a hyperactive immune state all the time. It also promotes normal, healthy lining and mucus of your gut, and without them, you actually don't develop a healthy immune system. So it's critically important. Then I'm going to mention one other thing, which I find literally mind-blowing, which is that it's not all about the digestive tract and about food. That there is a relationship between, it's totally validated, peer-reviewed, relationship between mood, stress, and maybe even things like autism that are going on in your brain that may be caused potentially by the microbes in your gut. So how is that even possible? Do they crawl up there and get in your head? No. So their vagus nerve is this nerve that connects your brain to your GI tract, and we normally think of it as regulating things like appetite. But the microbes can actually communicate with your brain through the vagus nerve. They also communicate through your immune system, which is floating around in your bloodstream and then communicates with your brain. So we're basically being driven around by all these microbes in our stomach telling us what to do. No, it's not exactly that. We're also controlling them. So depending on your mood or your stress or your psychological state, you can actually change your microbiome. So it's a two-way street. There's this communication through the vagus nerve and the immune system between the brain and the microbes that are living in other parts of your body.
Wow, that's incredible. That kind of overturns our whole self-awareness and what we think we're in control of. And
I think we should just accept we're an ecosystem.
We're never alone. Not have a nice place to be. Never be lonely again. So it sounds like this it's almost a fallacy to define humans as healthy or normal in disease states, because there's a lot of, almost a spectrum of different kinds of clinical syndromes that might be really defined by the community of the microbes and your interaction with those microbes that fill in that normal healthy state.
Yeah, I think a lot of people initially thought there might be a ground state or a normal state, and that anyone that deviated from that was unhealthy. And there's been, most of the microbiome research has focused on comparing people who have a diagnosed disease to healthy controls and asking what's different. And we have observed some differences, but maybe not as many as you might expect, or some of them have been, a lot of work to really justify. And the reason turns out that just amongst people that we would call healthy or normal people, there's incredible variability in the microbiome. So for sure, my 10 trillion are not the same as your 10 trillion. And everybody's different. So is it just random, the ones that you got? No. It's driven by a number of factors. Some of them are the same things that are driving your human genetics, where you were born, your demographics, your own genetics from your own parents. But some of them have nothing to do with what you acquired at birth, but to do more with what you did this morning, for example.
Like taking antibiotics.
Absolutely. Antibiotics yesterday or even earlier in your life, it might have been 20 years ago, will leave a signature. Whether you were born by a cesarean or a vaginal birth, whether you were breastfed or bottle-fed, things that might have happened deep in your past leave a signature. But what's interesting is you look at people who live in different parts of the world and they have a different microbiome. But then, it's a global world, we all travel, so someone moved and they come to San Francisco. Do they keep their microbiome from where they were from, or do they get a new microbiome? The evidence is mixed. So there are some signatures that may stay with you for your whole life. Like if you came from someplace with no routine access to medical care as a child, and you never took antibiotics, we could probably tell when we look at you. But more importantly, we can tell what you're eating, what drugs you're taking, how sanitary is your environment, various things like that. If you show me a sample, we might even be able to tell if it was taken in the morning or in the evening. So our own microbiome is just cycling day to day, hour to hour based on the inputs into the system, and yet there's this background of some kind of unique signature that's there as well. But the output of all of that is a huge level of variability. So amongst people who seem the same, who seem normal, if we look in their microbiome, they have very different bugs. And we kind of just have to accept that that's part of our genetic variability. And it makes it much more challenging then to assess what a sick state looks like if normal has so much variance in it. So that's one of the biggest challenges that we've been working on in this field of research.
Yeah, I think you mentioned something earlier about not only the diversity of microbes themselves, but the diversity of their genetic repertoire. And it sounds like health, or the state of being healthy or normal, is not just a matter of which microbes you may have and how many of each type you might have, but on a whole other level, what kind of genetic repertoire they might have to do what they do.
Yeah, absolutely. So what's been incredibly challenging is that the bulk of research in this field has focused, up until very recently, on describing the types of microbes that are there. It's basically the same thing that microbiologists or biologists have been doing, systemicists, as we call it, have been doing for a long time. I have this many of this kind, that many of that kind. And we can do that pretty accurately now. But what has come to light, and has been one of the major focuses in my lab at Gladstone Institutes, is that even if I have a specific species of bacteria, and you, by all measures, have the same species of bacteria, our actual bacterial genomes that we're carrying around could differ by as much as like 30 or 40% of their genes. So just knowing the types doesn't actually tell you what they're doing. So everyone's focused on who's there, which kind of bugs. Is there a particular bug that's associated with obese people, or with people who are at risk for a heart attack, or other questions like that you might be really interested in. And some really compelling stories came out. One of the most heavily touted, and everyone's probably seen it in the popular media, is that there's this microbial biomarker for obesity, that you have more of the firmicutes compared to the Bacteroidetes phylum. So what happened, though, was that was just not the case. That what matters is that what the microbes are doing. And when we went back in my lab, we analyzed the data from many, many different studies. We saw some really troubling things. That the amount of firmicutes in a person has more to do with which study they were in. It varies more between studies than between the lean and obese people within a study. And it depends what DNA extraction protocol the lab used, what statistical software for analyzing the data, all of these biases, what sequencing machine, all these really troubling things that we don't want to worry about can be driving it. So anytime there's a simple biomarker, like if you have this bug, you're going to get sick, and if you don't, you're going to get healthy. It sounds great. It's like drink white wine and you'll be fine. But it's not true, unfortunately. It matters whether they have bugs. Those bugs have specific genes in them. And a really familiar example is E. coli. Everybody has some E. coli in them. We all do right now.
We're used to hearing the term non-pathogenic.
Yeah.
Escherichia coli.
Right, it's non-pathogenic, and yet when there's one of these foodborne illness, food poisoning outbreaks, and you hear it with E. coli, you think, oh, well, anyone with E. coli is sick, but it's not true. The E. coli that caused that outbreak only have a few genes that are different compared to the E. coli that's probably in me right now and not causing any harm at all. So it doesn't help at all to know, oh, you have a lot of E. coli. You need to know, is it the pathogenic one or not? Right. And that's to do with which genes it has. And what's really troubling about bacteria, which I know you're familiar with, is that they lose and gain genes all the time. Not like us, we're pretty stable, but they are quickly dividing, and every time they divide, genes can come and go. Their genes can go onto these little pieces of DNA that just float around, get taken up by another cell, and eventually, in some cases, absorbed into their genome. So there's this horizontal gene transfer that we call it on these plasmids, and it is not random which genes get shuffled around, which genes get deleted, and which ones get added. They're really important ones. It's genes to do with things like the fiber metabolism that I was talking about earlier. So we could both have Bacteroides or a certain Clostridia that's supposed to be good for gut health, but if mine don't have the fiber genes, I'm in trouble. I'm still gonna get that thin lining in my gut. They're gonna give up on getting fiber, and they're gonna start eating the mucus that lines my gut. They're gonna start telling my immune system to go crazy. And that cannot be diagnosed by saying, oh, you have a Clostridia, or you have even a specific species of Clostridia. You need to know which genes are there.
So it sounds like you can almost dispense with getting bogged down in the details of which species are present and how many of each are present, and focus on gene markers. And would it be fair to say that a lot of the work you do has been really accelerated by applying computational approaches to looking for gene markers of disease or maybe signals of interactions amongst the community of microbes that might determine what makes us human?
Right, so I'm making it sound kind of easy. Forget about what kinds there are, let's just look at their genes. And maybe that sounds compelling coming from a genomics person. We have techniques, we can just go in and we can start looking at the genes that are there. But this is highly complicated. There are, as I said, maybe a few thousand types of bacteria, but there are so many, millions and millions of different types of genes. So until very recently, we just didn't have the ability to even assay the genes. We couldn't measure those. And that's why there's been this focus on the types. But a couple of technological advances have really changed the playing field. So one of them is in the area of DNA sequencing, that we can just simply extract DNA from a microbial community without having to cultivate those cells and throw it into a DNA sequencer, and for relatively little money, get a very deep genetic profile of the community. So let's think of a really concrete example. I want to study the microbes in your mouth. So spit in this test tube, Shannon, we'll just pretend. And I'm going to use my DNA extraction kit, and I get these pieces of DNA out of that sample. And there are millions and millions and millions of them, little pieces. We chop them up so they can be read by the sequencer. We do some chemical modifications, some molecular biology on them, so that we get what we call a library of different pieces that are about 100 or so base pairs long, 100 and so letters, A, C, T's, and G's from microbial DNA. And we can read those in a DNA sequencer. So this sounds good. Two problems. One, the size of the file that comes out of that DNA sequencer. So your little test tube of saliva produces a text file, which is literally just a big text file, string of A's, C's, T's, and G's. That one file would fill up about 10 laptop computers. That's challenging. So that's where the computing comes in. So we need these great DNA sequencers, but to make any sense of this massive text file, we need high-performance computing, which has advanced incredibly in the recent decades, so this research could never have happened without that. But also people who have the skills to come up with algorithms, statistical tools to mine that data and find the patterns in it, and that's what my lab at Gladstone does. So to just kind of give a real-world analogy, I think the text file is overwhelming and sort of makes the point of the challenge, but let me make it even more clear how hard this is. So we can think of the microbes as books. Okay, so you've got a lot of books, and what's a big pile of books? It's a library. So we have a library full of books, and what we want to know is the knowledge in that library. We want to know the books that are there, and we want to read the genes that are in those microbial genomes. It's like reading the books. Read the story. Read the story. I want the story of your microbes in your mouth. Right. So what do I have on which to reconstruct that story? The experiment that I just described is analogous to going into the library, picking a random subset of the books, not the majority, like a minority of the books, maybe 1% of the books or something, tearing a random page out of each book, throwing them in a big pile, so I don't know which page came from which book.
Maybe putting them through a shredder or
Maybe even shredding, and then asking what was the knowledge in that library. So thank goodness for really fast computers and the really smart people in my lab.
Right. Now, to take the story analogy one step further, stories are always constructed in a certain pattern. There's a beginning and a middle and an end, and there are heroes and anti-heroes. So you could probably use certain formulae to predict how the story might fit together. There are probably certain ways genomes are constructed. You could tell what, just based on the structure of the gene, what it might do. Is that true?
Yeah, so I have kind of two answers to that question. So one of them is yes, in order to interpret all these pages that are sitting in this pile, we do map back to what we know before. We don't throw all knowledge up till now out the door. We have thousands now, about getting close to like 10,000 genomes of microbes and non-microbes that have been sequenced, so we see the genes that are in them. So we can build a massive database of what genes look like and what genes that do certain things look like. So to analyze, we can reference back to this database. But then there's the challenge that I mentioned earlier, that only like 1% of what's out there has ever been studied, and it's mostly the pathogens and things like us, like monkeys and humans and rats and mice. And so we don't want to be biased too much by what's in our database, because we might miss something really interesting just because it doesn't look like something we've seen before. So yes, we don't forget all the books we've read before in that analogy. We use them. And frankly, some of the people in my field completely rely upon them because, okay, at least we know what we're talking about here. But we've realized that you miss a lot when you do that. And so our focus has been on trying to reconstruct the knowledge from ground zero, from those DNA sequencing reads, with reliance, but sort of minimal reliance on stories we've read before. And there is a lot there. So we have a project in my lab where we have identified gene families, things that you see consistently, like maybe in everybody you've ever looked at. Microbiome, it's there, and it's never been described before. And it's not like we have 2 or 3 examples, we've got thousands of examples of things that look like bona fide genes with important functions. Most likely, if we all have them, our microbes all have them, and we just don't know what they are. So it's pretty humbling to realize we really haven't read that many books yet.
That's amazing. I think one of the intriguing things for me, working as a researcher in this day and age, is that these new techniques also allow us to look for things, for new stories and new books that we've never read before.
Mm-hmm. Yeah, I think that there's this amazing ability now to look at things that can't be cultured and that haven't been looked at before, and it really opens the door because, frankly, the majority of what's there we don't know about. And I want to take your story analogy even a step further and think about characters in a book. So if these microbes are different characters, a good story isn't just about one character. What did that guy do? What did he get up and do? What did he do that day? What happened? It becomes an interesting story because he runs into other microbes and has his interactions, or maybe he runs into your human cells, right? So on top of the microbial complexity of like how many things there are, all the possible things they could be doing, how different it is between me and you, on top of that, there's the interactions between the different microbes. And these are across domains of life. It can be bacteria and virus interacting with each other, virus with host, bacteria with host, food coming in. So a huge level of the complexity are these interactions. And we're just starting to be able to look at those. They require following a process over time. So it's one thing to just say that 2 microbes co-occur. Okay, they're both in Katie. Well, that might mean nothing, unless you looked at a lot of people, or maybe even more compellingly, you looked at me over time. And so there have been a few great studies in the last few years who have looked at an individual person's microbiome every day for a year or two and looked at what happens and shown incredible variability, as I've described, but it's not random. There are particular components of the human microbiota, certain bacteria, viruses, archaea, microeukaryotes, that are co-occurring with each other consistently over time. When one goes up, the other goes down, and in some cases having what we would call a lagged response. When one goes up, then the other eventually goes up or down, but in a lagged response that suggests causality, that the increase in one is actually driving the decrease, say, in the other. A really interesting example of this would be there's a huge body of work on viruses that I'm sure you're familiar with that is emerging, showing that how we respond to a virus has as much to do with our own genetics and whether we got a good night's sleep and what medicine we took or other things like that as to do with what commensal bacteria we have. So your commensals are, in a sense, they're fighting the virus with you, and they can be on your side or they can be on the virus's side. And so in some cases, for example, whether someone progresses quickly to AIDS after an HIV infection has a lot to do with their commensal bacteria. Similarly, the viruses can be fighting the bacteria. So we think of viruses infecting us, but a lot of the viruses out there are targeting the bacterial cells. They're what we call bacterial phages. And they're, in a sense, keeping the bacteria in check. And so there's this complex relationship between the viruses and us, the viruses that infect the bacteria and their bacterial hosts, us and the bacteria, us and the viruses. So it's incredibly complicated just in the way that a really good book is, right? All these characters.
Right. So they're not only helping our digestive system and training our immune system and affecting the development of human beings over time, but they're actually setting up an ecosystem that competitively interacts and may protect us against invasive or pathogenic bacteria and viruses.
Absolutely. I like the ecosystem analogy, and I want to kind of take that and go in a different direction to say that not all the work in my lab is looking at the human microbiome. We're also looking at bacteria in natural environments. And literally there it is an ecosystem. And we're trying to take some of the ideas from ecology and then apply them to the human body. So we can literally think of the human body as an ecosystem and try to learn things by studying soil bacteria, ocean bacteria, bacteria that are on windshield wipers or air vents or floating in the atmosphere. And these same genetic techniques can be used to go out and sample seawater. Maybe all be familiar with Craig Venter and his yacht when he sailed around doing this, but there's a lot of other people doing it as well. Soils, the atmosphere, and these have amazing implications for things that we also care about, maybe as much as our own human health, like global warming. So one project in my lab is looking at what will happen to microbes as the earth forms. And what we're seeing is really interesting. Whereas a lot of macroorganisms, trees, large mammals, etc., are going to, polar bears are going to be threatened by global warming. We think microbial diversity in most parts of the globe, for example, most of North America, is going to increase massively in coming decades. They're psyched. They're ready. And so this is interesting in and of itself, but it also sort of begs the question of how does that relate back to us? And I remember you telling me an interesting story about microbes in deep sea vents that relate back to your work on human pathogens.
Right. Well, I think Craig Venter started the revealed the tip of the iceberg, but many have followed in sequencing the genomes of microbes and bacteria, basically, that live out in the ocean. They found the same virulence factors, the cassettes of genes that encode for virulence for Staphylococcus and Streptococcus and Vibrio cholera, many of the things that cause disease in humans, are in naturally occurring communities of bacteria and may be really important in the way that they live and survive and feed upon other host organisms in the natural environment, like plankton and other members. So these are themes that we've learned about in humans that then show up in natural communities of microbes.
So you're saying genetic material from a deep sea vent could be in my body right now?
We don't know how it got there. We don't know whether the same virulence factor in staph or the coccus and bacteria living in the deep sea vent came from the same source, or whether these virulence factors are out there and being traded like hockey cards between microbes to up the ante of their success in a community.
I think I remember that some of these real mobile elements often have the antibiotic resistance genes or the virulence factors on them. I think there's some mounting evidence that I've been hearing about at conferences recently that a lot of what's going on with antibiotic resistance has to do with our own taking antibiotics, but also what's going on in soil with soil microbes, that's the source for a lot of the antibiotic resistance, is bacteria kind of fighting each other out in soil. So it's all connected. I mean, it comes back to the human body, but
but it's out there in the natural communities we circulate in. And just to give a little shout out for viruses, they're often the mediators of this card trading habit amongst bacteria.
Small, but very important.
Exactly.
Wow.
So it seems like this one of the other things that we do in our lab is using these techniques, these new technologies, computational, but also in sequencing without having to know what you're looking for, to reveal new interactions between viruses and microbes in the mosquitoes. And it's a very powerful way of sort of revealing new interactions without having to know what you're looking for. So I was wondering if there are any sort of new questions you've been able to address that we couldn't have addressed in the past about what sort of defines the human microbiome in particular.
Right, yeah, that's a really good point, Shannon, about needing to know what you're looking for. So just a little bit of history on the field. It's not just recently that we were able to study the human microbiome. There were techniques decades ago for going and trying to explain what bacteria, for example, were present in the human microbiome. But to test if a particular bacteria was there, you had to know that you were looking for that bacteria. You went in with a probe, a molecular probe that would fish out pieces of DNA from a particular bacteria, and some of them were fairly universal and would pull out lots of different types of bacteria, but essentially you needed to have a plan of what you were looking for and base it on what you knew before, and then to analyze that data, you certainly had to map it back to previous knowledge, those books that you've read before. The thing that maybe I didn't emphasize enough in describing the sequencing experiment with all its complexities is that we don't have to have any idea ahead of time what we're looking for. When we sequence that saliva in your mouth, we will get whatever is there that might be, actually some of it will be you, Shannon DNA. Some of it will be food that you ate today that's the DNA of that is still in your mouth. There will be the bacteria that are native to your mouth. There will be the bacteria that were on the test tube where I took the sample, on the machines where I analyzed it. So if all I cared about was the bacteria that lives sort of under your gum or something, that is there, but there's a lot of other stuff there too. This is fantastic because we can address questions we couldn't ask before. We can look for genes and microbial taxa that we have no previous knowledge of. We can look for interactions between things without even knowing what they are. We can just say there's something, heck knows what it is, but you got a lot of it, and whenever your back is hurting there, you got more of that. So we can find those things without having to know anything. Then there's hard work to figure out the why. I'm not trying to downplay that, but in terms of discovery, there's massive potential. Unfortunately, there are massive complexities that come with that, which I alluded to in saying, okay, there's DNA on the test tube and maybe in the sequencer, answer. But it's not just about the sample collection. There's complexities, which I talked a little bit about earlier, but I just want to emphasize that relate to how you extract the DNA. The protocol for just getting the sample ready can enrich certain types of bacteria over other ones. And so if one study uses that type and then another one uses another type, you can get these systematic differences that look like, oh, people in Europe are different than the people in America, but it's just that you use different chemicals in the lab or different reagents. Then the machine that we do the sequencing on can introduce errors. They're not that frequent. These machines are pretty amazing, but even if you had like a 0.1% error rate, if you're reading, you know, millions and millions and millions and millions of letters, some of them are going to be wrong, and by chance, some of them will make one thing look like another thing. So that's a complexity. And then there's another one that is related to the fact that, you know, we have limited resources in terms of money, and also just how the machine works, the flow through it. So when I run your saliva sample, there's kind of a limit on how much reading of it I can do. And I don't even actually read all the DNA that's there. I read a random sample that's there. And so if there's a really complicated community in your mouth, there's a lot of different genes or a lot of different bugs there, I'm going to get a more shallow impression compared to if there was just one bacteria dominating that community. I would get to know a lot about that one bacteria. So when we as computational scientists come to analyze this data, it would be helpful to know, was that community complicated or not? And another subtlety is how big are the genomes, bacterial genomes, because of all this shuffling of DNA. The genomes can be big or they can be small. If they're small, then I get a lot more information about each one if I do a fixed amount of sequencing compared to if they're big. So let's say I wanted to see whether there was a particular gene that was associated with getting cavities. So if the bugs in your mouth have bigger genomes, it might look like you had less of that particular gene because I just sequenced it less. Or if you had a complicated community with a lot of different types of bugs, then for that one gene, I would just get less DNA. That's the facts. Right, but the actual amount of that gene, the concentration in your mouth, could be even more than in my mouth. But I just have a simpler community, and for a fixed amount of DNA sequencing, I get a higher number. So this sounds catastrophic. It's like, well, there's nothing we can do with this big pile. But amazingly, we're slowly identifying these different sources of error and coming up with methods we can use when we analyze the data to calculate, well, how much should I expect for you? Okay, I expect this much, but I got that much, then it actually is high compared to my expectation. But it's a lot more work, a lot more time on the computer, grinding away. So these analyses can take, on our supercomputer that has thousands of cycles going, the equivalent of like thousands of desktop computers stacked up, CPUs. We can analyze the human genome in like an hour, but it might take a few weeks to analyze your saliva sample. Yeah.
It sounds like one of the pitfalls of all this big data is teasing apart correlation and causation, and maybe sometimes making spurious conclusions about a relationship between a microbial signature and a state of health that may not bear out if you could really understand those interactions more deeply.
Yeah, this is what keeps me awake at night, actually, is worrying whether we can come to any firm conclusions. And honestly, the field is really hot. Everyone's excited about it. This fantastic audience is here tonight, and you know, we hear about these discoveries, you know, pick up the New York Times, it's there. And my fear is that our zeal over this potential for discovery is going to jump to conclusions very quickly. And then you'll read next week, oh, well, that wasn't true. Actually, we collected some more data, or we reanalyzed that data, and we found it wasn't true. And my lab has been a real pain for a lot of people in the field because we, as statisticians, feel sort of compelled to make sure that we're getting the right answer, and so we're reanalyzing studies that are coming out, and in many cases overturning the results when we look at the data more carefully. This is not always popular with my colleagues, as you can imagine, But it's super important because any person who hears, oh my gosh, I need to eat more yogurt or I need to eat less yogurt, they're going to run out and do that. And what if that was the wrong advice to give that person? I think we need to be very cautious and at the same time optimistic. I think there's great potential there. But the complexity of the experiment and the data analysis means that, frankly, I would say, you know, when I see a study come out, it's almost like flip a coin whether that one's going to hold up or not. So the field is new, and it's exciting. It's exciting.
Frontier.
Yeah, we're pioneers, but it's this phase where we're really we're just accumulating basic knowledge right now. What is the basic variability between people? What could I expect to find if I did this experiment? And so I'm super motivated to move on to these biomedical applications and health advice, but I feel like there's a lot of hard work and basic science and careful analysis that we need to do before we can jump to treatments and cures, unfortunately,
to really make the interpretation possible.
Yeah.
So you're working with the Gladstone Institutes as a research scientist. The Gladstone Institutes works a lot on humans. And so I wanted to know a little bit more about how you yourself, as a computational biologist working in an institute focusing on humans, got interested in dissecting the microbiome of humans.
Yeah, that's a great question. I have to say that it happened through phases. So I, like most of my colleagues, launched my career and started my lab based on human genetics. And in fact, the fundamental question that my lab was interested in was what makes us human. So we were comparing human DNA to chimpanzee DNA. The human genome was sequenced over a decade ago now, and a few years later, the chimpanzee genome came out. This is when I was getting started as a scientist, and it was incredibly exciting to compare humans to chimps and develop statistical techniques to try to find out what part of that human DNA is special. And that is still a component of my research program. But a colleague of mine, Jonathan Eisen, at UC Davis, sort of mockingly came up to me one day and said, you realize that most of your body is not human, and most of your cells don't have human DNA, right? Like this, what makes us human thing? Kind of a joke. I mean, you know, it's mostly microbes. This guy's very passionate about microbes. So, at first I kind of laughed and I said, yeah, I have some good grants, I'm doing fine, I'm working on my stuff. Good luck with that microbe stuff. And then, gosh darn it, he was right. I mean, it's just totally missing the boat to only look at the human DNA. And this is important in the question I was thinking about evolution and what you get from your parents and how we're different over time or in different parts of the world. But it has all these huge biomedical implications. So that was the first phase, with this kind of wake up that, like, as we started the program tonight, that we're not, most of the cells in our body aren't carrying around a human genome. The second step, I was still doing human work. I had some microbial projects that kind of gotten my attention. I had a couple microbial projects in the lab. I still saw them somewhat as a side project or an exploration. And in some of our work on the origins of humans, we collaborated with Carlos Bustamante and some of his colleagues at Stanford to look at people from Khoisan people, hunter-gatherers in southern Africa, that are potentially sort of closely related to the ancestors of all humans. They had saliva samples, coincidentally, since we've been talking about saliva samples tonight, from a whole bunch of these Khoisan individuals. And the student who's working on the project kept coming to lab meeting and saying, gosh darn, like 85% of my DNA sample is bacterial. I'm just throwing that out, I'm not getting enough data. Contamination, he called it contamination. And then we started realizing, wow, we could be looking at that. And then not long after that, great work came out of the Max Planck Institute and other places that looked at other groups of people living in sort of a non-Western lifestyle, the Hadza, for example, and pointed out the same thing that we discovered looking at these Khoisan microbiomes, is that these individuals not only are very unique in terms of their human genetics, they are carrying around microbes that you just never see in San Francisco. Lots of them.
Wow.
And for good reason. They eat different things. They have not had routine access to medical or dental care. Many have not taken antibiotics. And this tied into a hypothesis that many people have probably heard of and that I was aware of, but just the power of this kind of finally hit home for me. This idea of a hygiene hypothesis, that many of the diseases that are on the rise in the Western world, things like autoimmune diseases, obesity, metabolic syndromes, these may arise from the fact that we have such a clean lifestyle and you've used so many antibiotics, etc., have such a non-ancestral diet, eat something totally different from what our bodies were designed to eat. Our ancestors passed along to us genes for eating something very different than the processed food that many of us eat. And so, as a consequence, we may have lost touch with sort of the healthier ancestral microbiome. And not that these real modern people are fossils or ancestral, they're modern people just like you and me, but because they haven't had these exposures that the rest of us have had, they give us a little insight into what our ancestors might have looked like, and got me really thinking about the hygiene hypothesis. So then the third answer to your question of how I got into this was that at that point it became really compelling to me, being at a biomedical research institute where we look at HIV, we look at neurodegenerative disease, we look at heart disease, and I suddenly imagined there could be a microbial component to all of these diseases that if we're just looking at the human DNA, we're missing it. And then it also started to resonate with me as a patient. So I have 2 autoimmune diseases, and there are some human genetic reasons for that, but there's a lot about my health that is not explained to me by what we know in medicine right now. So I also just became incredibly motivated to try to understand my own health and the health of many other people who are suffering from these diseases that are on the rise. And logic suggests could really have something to do with the microbiome.
So I don't want to hug Katie all evening. It's now time to open it up to the audience for questions. So someone's going to come to you with a mic.
This question's in the front, right here somewhere.
Could you comment on the appropriateness of probiotics in the diet and give some sense of how to distinguish between different availabilities?
Yeah, great question. So it's about probiotics, how to tell if one ought to take them or not, and how bioavailable are the organisms in them. So I have actually, as a patient and as a scientist, I have great optimism about the potential of probiotics. I think that the microbiome is a massive, as we've discussed tonight, and unexplored target for medicine. And what we put into our body, dietary, drugs, supplements, etc., can change the microbiome on a almost hour-to-hour, certainly day-to-day basis. So, unlike your human DNA, which you're pretty much stuck with for life, modulo gene therapies and that may change that, the microbes in your body can be manipulated. You can add things, literally add microbes. You can try to remove them, for example, with antibiotics or other treatments. You can change the balance by putting microbes in there. You can put metabolites, so energy that would promote certain types of microbes, nutrients that would promote or stop them. So I think there's great potential. And however, right now the probiotic industry is generally not regulated. There has been a few do studies that have examined products that are available and tried to assess if they actually have in them what they are claimed to have in them, and even some of the most expensive or exclusive products often aren't what they say they are. I'm not trying to badmouth the whole industry. There's certainly, useful things out there, but I think there's a lot of work to be done still to ascertain exactly what the best way is to deliver a probiotic and even exactly what should be in it.
We'll take our next question halfway back on the very far right side of the orchestra.
What do we know now about the role of fecal transplants?
Oh, good. We can talk about food. I use saliva to poop. I use saliva as an example, but a lot of what we know about the microbes in the GI tract, not all of it, but a lot of it is from sampling feces. It's the easiest way to access what's in the GI tract and a pretty good measurement of at least the lower GI tract. Now, what is a fecal transplant? It's exactly what you think. It's taking feces from one individual and transferring it to another. A lot of the work on causality, we were trying to talk about causality earlier, a lot of the work on causality in the microbiome, having this microbe or having this gene leads to X, like weight gain, inflammatory bowel disease, change in mood or behavior, autistic syndrome, etc. A lot of that research is from studying mice and doing these fecal transplants. So either starting with germ-free mice that have no microbiome and adding microbiome in, either synthetically designed, so put together as a cocktail by a scientist, or taking the actual fecal matter of a, say, obese or a sick person and putting it into a mouse and see what happens. There is also a lot of interest right now in medicine and now also in industry, companies starting around the idea of trying to treat disease rather than through probiotics, through a special type, maybe you might even call it a special type of probiotic, a fecal transplant from a healthy person. For certain things, for example, infection, some of the colitis that's caused by a particular bacteria called C. difficile or C. diff that you might have heard of, can be treated fairly effectively, at least in some people, by a fecal transplant. What's really interesting, and this is very anecdotal, like a statistician is not telling you this right now, but there are a few examples now that these people who are cured by the fecal transplant lose their colitis symptoms, then pick up other characteristics such as being overweight or something like that, or metabolic traits from the person they received the fecal transplant from. So that needs to obviously be studied in more detail. Bigger sample size, please, it's like N of 1 or 2, but there's some suggestion, which I find incredibly intriguing, that some of the trends we've observed with mouse studies may hold in humans as well.
There's questions at the center.
Hi, thanks. It's really interesting. I have two different questions, if I'm allowed, really. I read that just recently that athletes presumably have a different microbiome than someone who's not an athlete. If you start working out, that it actually changes your microbiome, and just curious what you know about that. And the second one is, I've also heard about with genetics now, there's the epigenetics on top of just the genome, and studies about which genes are turned on and off, and I'm wondering if that plays into your research as well.
Wow, great question. So actually there's a whole group inside my lab, about a third of the lab is working on epigenetics. For folks who aren't familiar with that, that's the idea that you don't just need to read the genetic code, that the DNA can be chemically modified, which changes how you read it or how it functions. Or, in addition, the DNA is just not floating around as a big, long sequence of A, C's, and G's. It's actually wrapped around proteins, and those proteins can be chemically modified. Those modifications will give us even access to the genes or the sequences that turn them on and off to read them, and are really important in our understanding of not just which genes are there, but which ones are active right now. So most of what I talked about tonight is an experiment where we would just take DNA and read it. That tells you all of the microbes that are there and all the genes in their genome, but many of them could be dormant. So microbes can go dormant. They're not actively using their genes right now. Even the ones that are active might only be using a certain percentage of their genes, and that's true in the human DNA as well. This is the human and the microbes. So it's really important to start looking at RNA, which is a readout of which genes are active right now, proteomics, which sort of tells us about the proteins that are there, metabolomics, which tells us about what are the products being made by those proteins. Are you getting those small chain fatty acids from the fiber in your diet or no? So I think the DNA experiment is the most prevalent right now, but we really need to move on to all of these, including the epigenetics. Regarding working out, I know there's a cool study about the effect of working out on your human epigenome, and I did see that work on microbes. Seemed pretty solid, but as I mentioned, at this point I feel like we need multiple studies in multiple different places, validations, before you would go out and make a medical recommendation to people. However, working out has a lot of other good benefits regardless of your microbiome, so it's probably a good idea.
Our next question comes from the center of the upstairs balcony.
Hi.
Hello. I'm curious about how babies enter the world. Are they sterile in their mother's womb? And if they are, they must accumulate microbes in the first few years of their life. I guess I'm wondering how they do that. And when their microbiome reaches an equilibrium, maybe, you might say?
Yeah, the question was about babies. Are they sterile in the womb? Yes, as far as we know, the womb is a pretty sterile place, one of the few parts of the human body that seems to be pretty sterile. Birth is a major inoculation, whether it's a vaginal birth or cesarean. The infant is immediately exposed to a lot of microbes, and they are different in those two cases. As you know, there are a lot of yeast or lactobacillus, different kinds of bacteria that are present in the vaginal tract, and so if you're born by cesarean, you don't get inoculated with those, and so we can tell later in life how someone was born in many cases. However, it's not just the birth. You continue to acquire microbes throughout life. A lot of them are from a vertical transmission. Just like your human DNA, you acquire microbes from your parents. It looks like the evidence so far suggests more from mom than from dad, so you get half of your human DNA from each, but when it comes to the microbes, mom seems to play a bigger role. That is probably related to cultural practices and also to breastfeeding, in which women are doing more of the feeding and perhaps other types of care of infants that are involved in the early inoculations in the first year or two. What happens is amazing. So you go from sterile, this initial inoculation, you have a lot of variability between individual babies over the first year or two of their life, from day to day and between infant to infant. And it takes several years to establish what looks more or less like an adult microbiome. So we can tell how old an infant is very accurately, like to the month, essentially.
There's questions at your right, towards the front.
So I recognize that you're a statistician, and this is not going to appeal to that side of you, but I was wondering what the gossip and rumor mill is of the connection between helminth infections and autoimmune disease, especially considering the overlap that some immunoglobulins and eosinophils play in the role.
Great question. I'm actually excited about it. Even though I am a statistician, I'm also an evolutionary biologist, and helminths played a huge role in human evolution. So for folks who don't know, these are broadly parasites. And until very recently, and still in many parts of the world, we had a huge load of them. And it seems like the evidence is suggesting that they play a massive role, maybe even more massive than some of the bacteria we've been talking about tonight, in development of a healthy human immune system. And my reading of the literature is that loss of helminths may be a big component of this hygiene hypothesis, i.e. rise of autoimmune diseases, etc. Yeah.
Our next question comes from the balcony on the right side.
We're seeing a lot of these projects around the microbiome raise money on crowdfunding sites like Indiegogo, and I actually have a ubiome kit sitting at home. So I wanted to know whether you would recommend I even kind of go ahead with that kit. You mentioned a lot of complexities that we see, especially in the lab. Is it too early for these types of citizen science projects?
I'm a huge fan of citizen science, so no, I think you should go for it, and I think a lot of people should. Sample yourself, sample your door sill, sample the beach when you go down there, send that data in, it's great data. What I would explain to you about, say, the uBiome kit is this. As an analogy, it might be similar to what 23andMe or other ancestry companies are doing with the human genome. They don't give you your whole human genome. They give you what we call some markers, or places that commonly vary amongst people, and they tell you what variant you have at that position, and because of that, you're more likely to be from a certain part of the world. Same kind of thing, we're kind of at a similar stage with the microbiome. That kit is going to tell you for the common bacteria that occur in the human microbiome how much you have of each one. And that will be a personal signature. But it's, as we discussed tonight, just listing who's there, especially those well-known things, is kind of just part of the story. So know that there's a lot more there if you want to understand your human microbiome. It's not bad information, but it's a very sort of simplified or initial view of it. And know that if you take another kit and you do it tomorrow, you might get a different answer, so that your microbiome is changing. It's really dynamic, which is different from the human genetics. If you did that ancestry analysis a year later, you should get more or less the same answer, but I wouldn't necessarily predict that for the microbiome. So I think it's great that people are getting excited about it. I think it's a fantastic source of data. We're writing scientific publications based on some of the citizen science data. It's fantastic. And the assays that are being used right now by necessity, so they can be handed out for citizen science projects, just don't have all this complexity built into them.
This question's all the way at your left.
So you voice, I guess, some skepticism over conclusions found about like science around the human microbiome. Why would we study like the human microbiome versus more simple organisms, or like what are the advantages of each, and what are some of the things that we've found out about how these relationships work?
If I understand the question correctly, you're asking about why study this complicated community instead of picking a microbe or two that's really important, isolate them and study them in the lab? Is that the question?
Yeah, or perhaps organisms with similar microbiomes and their relationships, because all of these different organisms have relationships with their communities that live inside of them.
So why not study mosquitoes like Shannon does? Shannon, is it simple in the mosquito?
No. We're now starting to appreciate how many organisms invariably have a very complex holobiont, an enormous complexity of most microbiomes, especially in these enclosed chambers we call eukaryotic animals.
So yeah, maybe studying all the viruses that infect a single bacteria may be simpler. I don't know. There's a lot of viruses, a lot of phage, right?
And then really no bacterium can be really considered in isolation anyway. They typically live in communities, so to study something we've done cultivatable organisms and looked at their interactions with their viromes But I think we really do need to put those relationships in the context of the communities that they live in, and those are invariably complex.
So what you would learn from the simple system might or might not translate to what would happen in a fly or a human.
Right. Well, with the mosquito microbiome, which is very complex, there are bacteria, there are archaea, and there are fungi, and there are viruses, and the viruses might be mosquito viruses, fungi viruses, bacterial viruses, and they're all, it's almost like a set of nested Russian dolls. Parasites of parasites of parasites of parasites. No simple answer.
Too bad.
This next question comes from the right side of the orchestra in the far back corner.